Done.
Okay, hello everybody. So we are just missing a couple of speakers at the moment, but I think we shall begin anyway. So good afternoon, Kazakhstan, and good morning, UK. I am very pleased to welcome you to the 5th session of the 3rd Central Asian Research Forum on Sustainable Development and Innovation. Today's session will be on healthcare and sustainable development. My name is Nicola from Dundee University, and I will be the moderator for today's session. We will have 5 talks today, and then some time for questions and discussion. So I invite you to submit your questions using the chat option on Zoom or via the Facebook page. Unfortunately, our first speaker hasn't joined us yet, so we'll have a slight change in schedule, and we will begin with Mukhit, from a PhD candidate from Cardiff University, who will enlighten us on an option for vaccine delivery for the SARS-CoV-2 virus. Mukhit, are you ready to begin?
Thank you. Hello, everyone. Thanks for having me here. Let me introduce quickly myself, and then I will share my screen. So my name is Mukhit Kulmaganbetov. I am a medical doctor, ophthalmologist. Currently, I am doing my PhD in Cardiff University in vision sciences. I know this is not my topic that I'm going to talk about, but due to this relevance and due to the this idea that I have, maybe it can be helpful for someone who is working in this field. So let me share my screen. I hope you can see it now. All right. Do you see it?
Thank you, we can see that.
Okay, so the topic is SARS-CoV-2 virus, which is the etiologic factor of COVID-19 coronavirus infection of these days. And I'm not going to talk about how can we invent vaccine or treatment, but I'm going to talk about the transporters, especially nanotransporters for this vaccine delivery. You may accept this as a joke, you may accept this is a funny presentation, but this is what I can contribute to this world, what I can do, what I can suggest. Maybe someone can benefit from that. So this is just a proposal, idea. Hopefully you'll enjoy it and you'll have questions in the end. So as you know, yeah, first of all, I want to apologize that my video will be streaming from the bottom of my monitor due to this camera that's located in the bottom of my screen. So you may sometimes see my fingers or other parts of my body. I'm sorry for that. So let's quickly start. So the problem statement, the coronavirus COVID-19 has been recognized as pandemic by WHO, and from yesterday, the number of COVID-19 cases is more than 2 million, and then more than 134 deaths amounted today. So there are no specific antiviral treatment, no vaccine available currently. However, there are good news as well that from Nature, I just recently read that there are 98 confirmed COVID-19 vaccine candidates, 5 of which have already entered the clinical trials. Hopefully these guys can invent something and stop this pandemic and we can come back to the normal life. So as you know, this virus is has the envelope or membrane positive stranded to RNA virus with nucleocapsid. So nucleocapsid is the inner structure of this virus. This virus also is the largest known RNA virus and has ssRNA, approximately 30 kilobase in length and a structure of 5 cap structure and 3 poly A tail. Also, this virus is capable to producing an excessive immune reaction, what we call cytokine storm, and also it causes the extensive tissue damage through differentiation of B lymphocytes, which produces antibodies, especially interleukin. And also, one of the severe cases can cause the multiple organ dysfunction as well as the inflammation symptoms like fever and others. Also, what do we know about this virus, as you can see here. Hopefully you can see this part of this presentation. So in the center, you can see the nucleocapsid, the genetic material, RNA with N protein, and the membrane contains spike glycoprotein, M protein, hemagglutinin esterase, as well as the E protein. So what I want to suggest is to eliminate the central part and leave the whole outside part, so we can use these specific membrane antigens to use the targeted action of vaccine and therapeutic agents. These antigens can be recognized by TMP, RSS, and the ACE2 receptors presented in human body. And also, if we use for example, agents instead of this RNA, it can be any possible materials like enzyme, let's say protease, that can destroy the proteins of the components of this virus, or nuclease that can remove or destroy the RNA or nucleotic parts of this virus, or antibody which is already formed in infected people, or peptide that can block the expression of these proteins or replication of this RNA of coronavirus genome. So as you can see here, so this is the virus itself with the membrane and the nucleocapsid inside. This is the human cell on the down part. So this virus can be recognized by these receptors, and the endocytosis can happen. After that, the cell can be infected. So this is just the slide. How can the life cycle of this virus, this coronavirus, can be endocytosis and exocytosis. Between these two processes, replication and the transcription of the genome can happen after the entering. This is just like quick background information about general virus lifestyle. So after they entering into the cell, release of viral genome can happen, and the ribosome can translate the viral polymerase. After that, this viral polymerase can replicate and translate the viral proteins, and then replicate the genome RNA of this virus. After that, after the translation and the forming the nucleocapsid, it can go outside of the cell, or else we can call it exocytosis, and these viruses can go to other cells and infect other cells, and this life cycle can repeatedly happen. So what we can see here, what I want to suggest is to remove this RNA from the virus and implement this agent. As an agent, I suggested some materials before. We can call this membrane agent structure. This membrane agent structure, you can see everything outside is the same, but the only difference is the agent inside instead of RNA. It can go to the already infected human cells, again recognized by these receptors. After that, this infected cell the endocytoted these membrane agent structures, and then release of these agents will go to the cytoplasm and destroy the virus itself or viral components. So after that, we can get the healthy human cell. So what are the benefits of this methodology? Is the targeted transport of treatment and preventive agents, also the exact localization of this effect, also the reduction of toxicity. Let's say we use some protease or nuclease that can also kill human cells or human proteins or human DNA. In which case, possibly using the membrane of virus will reduce this toxicity, and we can reduce also the side effect, not saying eliminating, but possibly reduce. Also, we can reduce the dosage of these agents, as we can say these agents will not go to the all cells or will have the systematic effect, only will go to the cells that already infected. And also because of this, it can be cost effective. So also there are several types of the viral vaccine development. in medicine, we can use live attenuated or whole inactivated virus, or else some particles like synthetic peptides or DNA or RNA or recombinant subunits. These also particles can be implemented into the viral membrane and also can play a major role in vaccination, not only the treatment that I already demonstrated before. So these are the only suggestions. Hopefully, because of this large size, we will be able to implement almost any macro or micromolecules that we can do that. Of course, it should be considered by biochemistries, biotechnologies, considering the size, also the other chemical and the physical properties of this viral membrane. As this is the proposal, you may ask about the funding. Unfortunately, due to the lack of time and my lack of my experience in this field, it hasn't been calculated, but I'm sure it will be calculated if someone will be interested in that. So first of all, we have to ensure the dialogue and the communication with potential partners, and then they have to estimate the amount of funds for the capacity of the viral laboratories in Kazakhstan. And I'm not excluding UK as well, as I study and work there, and the access to the reagents, chemicals, and also team and size and other expenses. Thank you for your attention. This is the very quick and short suggestion that I want to suggest. If you have any other questions, please feel free to ask now or later. Thank you.
Thank you very much, Mukhit. That was a very topical talk. And so please continue to submit your questions. Our next speaker will be Yerlan Ramankulov, the Director General of the National Center for Biotechnology and Associate Professor at Nazarbayev University. Yerlan will also be talking about a novel approach to fighting coronavirus. Thank you, Yerlan.
Hi, everybody. Let me put the on the screen my presentation. Just a second. Okay. So, can you see it?
Thank you. We can see.
Okay. So today I would like to tell you about our efforts to make a coronavirus vaccine, and we were involved in this activity after we have done the very nice, unexpectedly nice real-time PCR system for detection of the viruses, the coronavirus. And we had to make this vaccine because when in January, back in January 2020, the sudden urge to be able to detect coronavirus in people suspected for coronavirus infection appeared. The Ministry of Healthcare came to us asking if we are able to come up with such a detection system. In about 6 days, we made upon the WHO specifications our PCR system, which turned to be quite sensitive and good. It was actually better than the Russian system, which was widely used in February and beginning of March, which was able to detect about 1,000 particles per ml of the sample from patient. Our was doing less than 200, and so now we made a second generation of that, which has it's a multiplex system which can divide distinct between the general coronavirus and the COVID-19 per se. Plus it has a number of the internal controls which control purification of RNA, the reverse transcriptase reaction, and the reaction real-time PCR reaction itself. So, when we got this, the government, and we were only people in Kazakhstan who were able to do that. We produced about 11,000 test systems for our healthcare system, and that it's used. The Chinese and Russian ones are used for widescreen, the large-scale screening, and our system is used for the as a judgment call when the results are not distinctive or complicated by the something which makes them not very easily interpretable. So now we were asked to come up with the vaccine, and along with the Institute for the Biosafety and Biosecurity Problems in Atar, we are going to make 2 candidate vaccines. And the one of the problems which people, actually, let me say a couple words about the vaccines we're going to make. Here you see very familiar, unfortunately for everybody now, image of the coronavirus, and you probably know that the proteins which are exposed to the outside of the virus particle, first of all, S-spike glycoprotein, which has the probably strongest antigenic profile. Also, the okay. So also membrane glycoprotein M and the small envelope protein E. Also the let's see, and the now out of 78 candidate vaccines, which was mentioned by the previous speaker, about half of them are subunit vaccines, about a third of them are vector vaccines, and we're going to just do just that, do 1 subunit vaccine and 1 vector vaccine, because it's the most prospective and popular method, and popular for a reason, has the highest potential for success. And when one produces, for example, a subunit vaccine, so the big problem is that the gene spike protein S is fairly large protein, which is about 1,200 amino acids large, and to express this protein in the native conformation is a very challenging thing to do, especially with the fact that this is like a protein, not just a protein. And expression of proteins in the industrial conditions are normally done in the E. coli or in yeasts as the cheapest and the easiest way to produce biomass. But the fact that it's a glycoprotein is making it making the possibility of the producing irrelevant piece of protein fairly visible and real. So in order to, if production of the protein will show that protein is not conferring a immune response because of its not native conformation, one has to have a plan B for production of the designed protein or fragment of a protein. In this case, we actually, and most of the vaccine designers and producers are actually opting for the subunit. Let me see. Yes. So here is a spike S protein. It has a the part which is embedded in the membrane, and the part which is but connected to the receptor of the ACE2 receptor of the human cells, and it's called receptor binding domain. And basically, in most of cases, the strategy is to take this part of the protein, which is easier to express and purify and present to the immune system in this native conformation, rather than whole protein, taking into account that the part of this is very hydrophobic and embedded into the membrane. So the way to deal with this is to express it in the eukaryotic cells. And as everybody probably knows, the eukaryotic cells are very expensive and hard to grow. The biomass is fairly low in respect to other methods with fast-growing bacterial or yeast cells. Yeast cells, also being eukaryotic, do not provide the adequate, very often, glycosylation, because mostly mannose, and in our case, it's a let me see, has more heterogenic glycosylation, and so the glycoprotein folding is better be done in more higher evolved cells, such as plant cells. Why plants? Plants are much easier and faster growing things rather than eukaryotic cells in culture. And lots of companies, you can see here, produce the vaccines into the plant cells. Here are the antigens for different pathogens, for different viruses, which are successfully grown into maize, potato, spinach, the tobacco plant, Nicotina benthamiana, in rice. And this is also a nice approach when you need there is a search for the crisis, in crisis situation, we need a lot of antigen to produce lots of vaccine. So this is a nice way to get these antigens if you have a way to immediately recruit some plantations, for example, of potatoes or tomatoes, and treat them in a quick way, making them produce your protein, which is a vaccine antigen or antigen for test systems, whatever you need. And a way to do it is to get the plant virus, and here you see the tomato bushy stunt virus, which is totally harmless for people, and that makes it much easier to work with, where we can substitute, in this case, capsid protein, which is not essential for infection, with the protein of interest, and infect the plants with this virus, which makes plant to express protein of interest. And then you harvest the biomass, lysocells, or grind the tissues and lysocells, and then get the protein of interest. So it's a rapid, very scalable, accurate, and versatile method. And as I showed in the previous table, used by a renowned vaccine producers, Here you see the genome of the tomato bushy stunt virus, or TBSV, where we have the adapter with the number of the restriction sites, where we can put the proteins of interest. Here you see the fluorescence of the green fluorescent protein being transiently expressed in the, this case, tobacco plant. And it takes about 3 days from the moment it has been sprayed upon the plants, and you get a very nice expression of the protein of interest. So this is the one of the branches which we're going to take. Here's the actual, you can see here a immunoassay, enzyme immunoassay for qualitative detection of bovine leukemia virus in 96-well format. The ELISA assay, and which we designed and produced. And here we, the bovine leukemia virus antigen was expressed exactly according to this procedure and method. So, and this is approach which we're gonna take as a plan B. So we didn't get to end with the plan A naturally. So, but if anything, this is something we're gonna do, or actually we're gonna do it anyway. And so we'll clone the RBD of the spike protein S into the tomato virus and express. So this is, I guess, all I'm gonna show today, and I will be happy to answer your questions.
Thank you very much, Yerlan, for such an interesting talk and very relevant to the current situation. I'd like to remind our participants that there will be an opportunity for discussion with our speakers towards the end of the session, so please continue to submit your questions.
Thank you.
Our next speaker today is Rauan, who is a neuroscience PhD candidate at University College London.
Hi, thank you, Nicola. Yes, I can't share my screen before the previous.
Yerlan, could you stop sharing your screen, please?
Yes, just a second. I'm trying to get back into the, let me come out of the Zoom and I'll come in again because I have some technical glitch.
Hi, can you hear me?
Yes, we can hear you and we can see your screen. Thank you.
Thank you, Nicola. Probably I have to do it like this. So hello, everyone. Today, I'm going to briefly present a project proposal that we have named Establishing Human Genome Variation Database for the Genetically Appealing Central Asian and Transcaucasian Conference. Along my presentation, I will shortly explain why the genetic studies in Central Asia and Transcaucasia are appealing and why it's important to have a reference human genome for this region. Central Asia is made of 5 countries, including Kazakhstan, Uzbekistan, Turkmenistan, Tajikistan, and Kyrgyzstan, whereas Caucasian countries are made of Azerbaijan, Georgia, and Armenia. The overall population of the region is around 86 million, and the population in these countries is extremely diverse. Each country has a unique history and culture, and the genetic diversity is the highest in Eurasia. High rates of consanguinity in Azerbaijan, Turkmenistan, Uzbekistan, and Tajikistan enrich the region with rare genetic disorders. As you see on this map, the region resides right in the middle of Eurasian continent and played an important role as a natural corridor for various early human migration routes out of Africa. Early hominins and modern humans migrated from Africa through Central Asia and underwent Paleolithic maturation phase here before spreading across the world. Later, Indo-European people emerged from the Caucasus region and populated whole Europe and Central Asia. Then this region was occupied by Turkic people originating from South Siberia. And finally, there was a large admixture with Mongolian people due to their invasion. As a result of this demographic history, a unique and diverse Central Asian population has emerged, some ethnic groups still preserving European phenotype. And the presence of Western and Eastern mitochondrial DNA was found in Central Asia. Unfortunately, little is known about the genetic background of many disorders in this region. Therefore, launching genetic studies is very important. With this in mind, back in 2017, we have established the University College London Central Asia Trans-Caucasia Disease Diversity Project. Disease diversity means that many human disorders differ in terms of their genetic causes and clinical presentations depending on the ethnic origin. Therefore, studying diverse populations is very important in understanding the genetic structure of many disorders and designing personalized treatment. Thus, from 2018, we have started establishing research collaborations between UCL and various medical and research organizations in Central Asia and Trans-Caucasus. The UCL team of neurologists and neurogeneticists have visited almost all the countries in the region, excluding Turkmenistan. We have met key collaborators from the region, personally recruited patients to our project, and delivered many lectures on neurogenetics. To this date, all logistics is managed, and we have received more than 5,000 blood samples from very rare genetic undiagnosed neurological disorders from the region. We have provided them with free genetic testing, mainly exome sequencing. We also have found novel genes in Central Asian population that are very important for understanding of human neurobiology. So, what seems to be the problem? Well, unfortunately, only about 10 to 15% of the patients could reach molecular diagnosis from Central Asia and Trans-Caucasus, whereas in European populations, this figure could reach around 40%. The reason why the diagnostic yield is low in Central Asia is grossly in the absence of human genome variation database. What is human genome variation? Each individual is different from others by the sequence of his nucleotides in his DNA. His DNA sequence is close to other individuals from the same ethnic group and could be largely different from an individual from other ethnicities. Some of these DNA sequence differences could be associated with a rare disease. We call these sequences as mutations. But if one sequence is considered as a mutation in one ethnicity, in another population, the same sequence could be just a benign variation, not associated with any disease. Therefore, in order to understand the consequences of different nucleotide sequence variations, the Human Genome Variation Database was built to estimate the frequency of those sequence variations in certain populations. This helps us discerning rare from frequent DNA sequence variations, where rare ones could be associated with a certain disease. So on this map, you can see that the current human reference genome was built mainly on European populations. And this European bias leads to poor disease risk prediction across global populations, incomplete understanding of the genetic architecture of human diseases, and more importantly, it worsens health inequalities, as European population will have access to molecular diagnosis, prevention of genetic disorders, and personalized medicine, whereas other undiscovered populations will not have this opportunity. From our exome sequencing, we find many single nucleotide variations with unknown significance that limit our interpretation of the data. Similarly, many people from Central Asia and Trans-Caucasus countries send their blood samples to private genetic labs in Germany, Russia, South Korea, pay around $1,000 American, but usually get negative results, as those private labs face the same problems as we do. They cannot interpret the significance of many DNA sequence variations from our population. Therefore, establishing human genome variation database on healthy individuals from our region is very important. Since our UCL Central Asia Trans-Caucasia Disease Diversity Project has made a good background for human reference genome project, in Central Asia and Transcaucasia, we make the following project goals. So we want to start building genome variation with exome sequencing, as it is cheaper than whole genome sequencing and could cover the protein coding sequence. We also want to provide educational activities at UCL to those people involved in populational genetic studies from Central Asia and Transcaucasus. The project results should be freely accessible online. We need to then continuously enhance the database, not only with exome, but also with genome data. And finally, we want to improve genetic data interpretation for research and clinical purposes. We think that it's reasonable to start the project as a pilot, sequencing 500 healthy people from each country in Central Asia and Transcaucasus. We will recruit healthy subjects according to the unified study protocol. Then perform exome sequencing and bioinformatics. And later, we will make a web platform to access the database with further enrichment. We have agreements with world-leading genetic labs and can make good coverage one exome sequencing per 106 pounds per sample, which is very cheap. DNA sequencing for us will cost around 13 pounds, and for a target of 500 samples from each country, the pilot will cost around 478,000 Great Britain pounds, which is per one country. For one country, is 59,000. Our project is supported by UCL Queen Square Genomics, Genome England, the American Broad Institute, and Columbia University. These organizations can provide with free exome sequencing, but only for patients. Whereas for this project, we need to study healthy people. We also have support from the big genetic labs like Microgen and LGC, and collaboration in place with key researchers from Central Asia and Caucasus. To conduct the project, we need a financial support from local governments and organizations and international organizations. So the expected benefits of the project would be yielding many novel disease-associated genes from the region, delivering molecular diagnosis to unresolved cases, and providing genetic counseling and personalized medicine, and improving the research potential in Central Asia and Transcaucasia, which is also very important. Thank you very much for your attention, and I would be happy to answer your questions.
Thank you very much, Rauan. It's very interesting to hear about the complexities of human genetics and things which are often overlooked. Our next speaker today will be also a neuroscience PhD candidate, but at King's College London. Daulet will update us on the progress and challenges surrounding neurodegenerative diseases. Daulet, if you're ready. Daulet, please don't forget to unmute your microphone.
Did you see the presentation? Sorry about this.
We can hear you, but I can't see the presentation yet.
Okay, 1 minute. Try one more time.
Thank you. Excellent. We can see it now. Thank you very much.
Okay, thank you very much, Nicola. So I try to be brief and save the time of all our colleagues and listeners. So my topic is 1 minute. Okay. My topic is neurodegenerative diseases, progress and challenges. Generally, my topic will consist from 3 parts. First will be introductions, the relevance of topic. Second will be more about general concept of neurodegenerative diseases, how it's classified, and what is the basic principles of classification of neurodegenerative diseases. And third one will be a little bit about what I am doing, what I am investigating, and some outlook, and maybe a little bit how it could be used in a clinical medicine. So, okay. Little bit numbers. As predicted, United Nations in the next 30 years, the human population will rise on 2 billion of people, and this increasing in number will be due to the aging of people, not the increasing the birth rate. As you see here, the growth rate, the left axis, it's a declining. while the people who are living longer than 65 and plus will come in more and more, and the proportion of this group of people will dominate on some point. So in 2050, there will be 1.2 billion of people whose age 65 and above, and according to the World Health Organization statistics, about 25% of elderly people will suffer from neurodegenerative diseases. And unfortunately, nowadays, we don't have any treatment, any cure, and at the same time, we don't have any diagnostic tool. Because the problem, I'll talk about this a little bit further, that we are diagnosing it when the patients come in our clinics, and we can't even slow down it. It's just symptomatic therapy. We're just making the life of patients a little bit easier. At the same time, we are not considerably helping them. Another diagram, another figures is, as you can see, the 65 plus people ages, they are increasing, and the rate is very high and higher than other groups. And another thing that I want to pay your attention is that people who is older even than 80, they will increase the increasing rate will be even higher than 65 plus. And in the absolute numbers, it will be from 143 million to 426 million. In this figure, I want to pay your attention that this is not problem only developed countries, countries with good quality medicine, high quality of life, you know, all this stuff. It will be problem of whole country, all countries, even developing. And you can see that, for example, in South America, there will be dramatic change in the proportion of 65 plus age people. And our Kazakhstan also increasing this group of people. And it means that in the next 30 years, the whole 5 continents will face this problem. No, second part is about neurodegenerative diseases. It's a group
Daulet,
of diseases.
excuse me, Daulet, sorry to interrupt. Your presentation has frozen, so we're unable to see your slides.
Oh, really?
I'm afraid so.
Okay. Which slide you are now?
The first slide.
First slide, okay. I don't know what's happening. Is it moving now?
It has still frozen.
Okay, I'll do it without slides. I'm sorry, I'll do it without slides. Hopefully it will just, you know, it's a
catch up.
side effect of this kind of conferences, I think. Yeah, so about neurodegenerative diseases, this is the umbrella term which includes lots of clinical diseases, and the main definition of this group of diseases is the effect on a neuron of the neural cells of central nervous system. And the common denominator of this group of diseases, they all have protein nature. I mean, one of the classification, if you look from the side of neuroscience, it's the accumulation of pathological protein aggregation and leading to the cell toxicity, and at the end it is leading to the cell death. So one of the approach to classify this group of diseases is to use this classification, and we will have 4 big groups according to these proteins. First group will be beta amyloid. The famous representative of this protein inclusion will be obvious Alzheimer's diseases. And you are not seeing my slides still. Yeah, am I right? Okay. So beta amyloid, it will be Alzheimer's diseases. Second group is the tauopathies, where the tau protein, the reason of all the disease. Third group, sorry, is synucleinopathies, and it represented, for example, 5 clinical diseases. It's a Parkinson's disease. It's the second after the Alzheimer's disease. Dementia with Lewy bodies is the second reason of dementia all over the world. It's multiple systemic atrophy, poor automatic failure, and Parkinson's disease with dementia. And the last group is the TDP-43 parties. And for example, the famous representatives will be amyotrophic lateral sclerosis, frontotemporal dementia, and the common denominator even of these all, it looks like diverse type of proteins. They all share one distinguishable feature. They all have a special amino acid sequence, which is called low complexity domain, which can to aggregate, to accumulate. All these proteins which I listed, beta amyloid, tau, synuclein, TDP-43, they are conducting the physiological function. But on some point, it's become pathological one. We don't know why. What's the trigger? What's happened? But we know that on some point they change in a pathological form, start aggregating and killing the cells. And here I want to move what I am doing. In this whole group of diseases, we are focused on a synucleinopathies. It's a Parkinson, dementia with Lewy bodies, and as you remember, it's alpha-synuclein, the reason of these diseases. And another member, which we suppose play a crucial role in this group of diseases, and not only this, but all neurodegenerative diseases, it will be choreofurin. Choreofurin is a protein which is shuttling the proteins, RNA between the cytoplasm and nucleus, and vice versa. And it plays a crucial role in a nucleocytoplasmic transport. So we are investigating, and this is our crucial question. Maybe the choreofurin is not playing the classical function, just shuttling the proteins between the cytoplasm and nucleus or chorioplasm. It may play a new role, chaperone function towards these, for example, synuclein. At least in 2010, one of the prominent researchers, they already proved that choreofurin plays essential role toward TDP-43, and it prevents creation in soluble forms of TDP-43, and as a consequence, it prevents whole diseases, even slow down and can hamper it. So, okay, and this is what we are doing. We are investigating choreofurin, the role in with alpha-synuclein. We already get some results, as we already find out that it's already colocalized with alpha-synuclein. And my supervisors conducted pilot study before my PhD with post-mortem tissue of patients, which I'm going to make it bigger nowadays, to make a more investigation in this field. They find out that even patients with, for example, DLB or PDD, they will find out the colocalization. It means that choreofurin try to protect alpha-synuclein, try to prevent the aggregation and accumulation. I mean, it's a hypothesis, but we're already getting the information about this, and now I'm chasing this approach, this idea, and perhaps if we prove the role of choreofurin in the future, it could be the pharmacological target to manipulate, which could, for example, slow down or even hamper the whole pathological diseases, and can be a solution of not only synucleinopathies, but lots of neurodegenerative diseases. And here maybe I'll finish, but before I finish, I want to say thank you for my fantastic supervisors, Dr. Frank Hurd, Professor Dag Arslan, Dr. Tibor Hartabagi, my funding scholarship, our governmental scholarship, Bolashak, the support in my PhD journey. So thank you very much for listening. I'm sorry for this technical inconvenience. Anyway, if you have a question, I'm more than happy to answer.
Thank you, Daulet, very much, and for continuing despite the minor technical difficulties that we faced today. I hope that there will still be plenty of questions during the discussion session later. So our final speaker of this session is Galimjan, a neurological surgery resident at the International Neuroscience Institute of China and Beijing Institute of Functional Neurosurgery. Ganamjan will discuss the development of space medicine in Kazakhstan. Thank you, Ganamjan.
Hi, Nicola. Can you hear me?
Hello, we can hear you.
Yeah. What about presentation? Can you see?
Yes, we can see your screen.
Yeah, you can see. Wait a minute. What about now? Is it okay?
Yes, you're in presenter mode, so we can see your notes as well.
You can see this one. Yeah, I will just take in a bigger mode. Yeah, with how about now?
This is good. Thank you.
This is good. Yeah. First of all, thank you for invitation. I hope all guys are safe. I have some points actually. Can I use by this one? Yeah. First of all, thanks for invitation. If there is some presentation, you can just say to me. I will take it in a Zoom. Actually, today I'm want to explain a new field that maybe will be interesting to medical students and especially in other professionals. That in a future perspective, it will be a very relevant topic. And what about my voice? Is it okay? Can you hear me?
Yes, we can hear you clearly.
Yeah. And I hope all guys are safe at home. Stay safe. First of all, I won't say there's not of conflict of interest and I have nothing to disclose. I will introduce myself because commission asked to give some introduction. I'm neurosurgical resident and Peking Institute of Neuroscience or Functional Neurosurgery, Neuroscience and International Neuroscience Institute of China. I'm interested in functional neurosurgery, aerospace medicine, especially effect of gravity and microgravity on intracranial pressure. Passionate about epilepsy, microgravity research, especially surgical therapy and cognitive behavioral issues. This is my overall. And what I wanna today talk about, first of all, give some brief history about space exploration. Secondly, physiological changes of space travel. Third of all, innovation of cluster of Nazarbayev University. Give some opportunities to Nazarbayev University students, research opportunities, and development and recognition of new specialty, opportunities to participate in space activities. And first of all, as we know, history of space exploration has been accelerated even since Galileo developed his first telescope, pointed it towards the stars some 4 centuries ago and published his finding in a small book titled The Starry Messenger. Since then, new and ever important technology was invented and enormously creative astronomers, astrophysicists, and space scientists have learned and made exciting discoveries about our vast surrounding and extraordinary places which we live. This picture, look for this picture taken from Hubble telescope. It's about 30 years to Hubble, today's birthday of Hubble telescope. And at that time, this was interestingly, one guy offered interesting idea. It was the last 70s, about 70s years. At that time, there was interesting ideas that Jared O'Neill offered that could we become an interplanetary species? And what's the next about space travel? And his idea was about, is the surface of planet really the right place for expanding technology civilization? And his idea was really interested, but a lot of people, especially in America, and to become his idea to become a true, he was used, and he said that about more important than material issues are opening of new and high frontiers will challenge the best that is in us. The new lands waiting to build in space will give us new freedom to search for better government, social system, and ways of life. And his ideas was very interesting. And At that time, as we know, Soviets and America was researched in deep space exploration team. And you can see this presentation, it's a new presentation about this year and after 10 years, but at that time, 17s, they have this kind of idea that go to the moon and after the moon go to the Mars. But unfortunately, Hollywood replaced this idea because Hollywood's movies is much cheaper than going to making moon exploration and Mars exploration missions. And we just forgot this kind of idea and vision. And let's talk about aviation and space medicine. First of all, what is this? This is a study of all factors affecting the human body in the flight, in health, as well as sickness, as a means by which those flying may be protected against potentially harmful effects of the abnormal environment. Let's talk a little bit simple. Aerospace medicine is a preventive and occupational medicine specialty that promotes health and functioning well-being of pilots, air crew, astronauts, and as well as the passengers. It's involved to study of biological, psychological effects of aviation, including exposed to changing temperature, large inertial forces, oxygen deprivation, air sickness, and fatigue. And if you're interested to be a professional in this field, you have to accomplish several specific competencies. First of all, you have to know about regulation and structure of international bodies. As you know, yes, as you can see from this presentation, there's a lot of regulation, global aviation and global space regulation. You have to know about them, and you have to master a level of diploma in aviation medicine. Secondly, you have to know about all clinical presentation of common diseases, not only on the ground, but also in the environmental conditions. And about hypoxia, you have to know about hypoxia, how to train hypoxia. You can see there's the chambers, barocamber chambers, and there's the pilots which will training to different altitudes, about 1,000 feet, 2,000 feet, and more. And you have to know about accelerational forces. As you can see, there's an example, there's centrifugal equipment and giving you training. Later I will explain about that, and about accidental investigation, equipment integration. You have to know this mask, how it works, how oxygen complies, aeromedical evacuation, thermal, human factors, noise, vibration, spatial disorientation, vision, and space medicine. You have to know all physiological aspects that human will take in space. You can see there's another picture. They're making CPR in a space. It's totally different that we're making. And there's some videos I want to say about accelerational forces. For example, one of the acceleration forces is the G forces. Centrifugal force. And what's the G-force? And if you say simply, I can describe that weight per unit of mass is pushing on you. It feels like, as I said, weight is pushing on you, every part of you. So that for fighters, pilots in training have to spend some time in centrifuge that we see to prove they can handle this kind of pressure on military forces. This is about G-forces. And let's look. And you can see from this presentation, what about my voice? Can you hear me?
Yes, we can hear you.
Yeah. And about G-forces, you can see from this presentation, astronauts and also experiencing G-forces. You can see from there G-positive forces that impacting on his physiological a lot of systems. But as we see from a previous video, there is the smallest time of impact is not giving us huge impact on your physiological changes. And from this presentation, you can see that pilots experience negative G-forces, positive G-forces, and backward G-forces. For example, let's say about the positive G-forces. As you see from this presentation, there's the heart. A heart is pumping blood to the brain to ensure the normal function of brain. But when the force is pushing forward down, heart is just hard to pump this blood out. But if you will change the direction of this force, it's a little bit easier. And this brief introduction of G-force you have to know as aeromedical specialty. And let's talk about some things about astronauts that experienced some physiological changes in space. As we know, there's a lot of doctors. We every day handle a lot of patients on the ground. And with simply taking from pathological condition to and convert them to a healthy condition. But when it's about pilots, we're just taking from healthy condition and putting over unhealthy condition, space condition. So it's really risky. And let's talk about some physiological changes of astronauts in space. If you will divide, there's short-term physiological alteration and long-term physiological alteration. And first time when astronaut and all person in a space experience a kind of puffy pace and phase congestion because of microgravity and a lot of forces that already known, not same as in the ground. And you have to experience this kind of feeling. And also there is vestibular changes. You can see from there, back pain, anxiety, and there's long term as you have to experience vision problems, orthostatic system problems, bone density loss, neurolithiasis, kidney stones, radiation-induced changes, and muscle deterioration. And let's talk about this International Space Station. They also experience, they have a gym, but a little bit different gym. They have to run every day to prevent the atrophy of muscles and skeletal system. And you can see there's a kind of dumbbell they're making every day to ensure the safeness and functional and ensure the system is doing well. And let's talk about immediate changes that a person experience on a space. First of all, the neurological, they experience headache, motion sickness, visual disturbance, neurological differences like kidney stones, most frequent problems, and psychiatric, not studied well, but it's also interesting topic about cardiovascular changes as arrhythmias or orthostatic changes, as I said, in G-forces, orthopedic changes like space osteopenia, trauma and infection disease. As you know, space station have a lot of bags that have to clean every day. And let's talk about long-term changes. Interestingly, there's Times published in 2005, Scott Kelly, astronaut, he spent 1 year in space and he experienced this space environment on his physiological changes. And you can see what we will experience in long term. First of all, is cardiovascular reconditioning, immune system suppression, psychiatric issues. As you know, psychiatric, you're going to another planet for maybe staying for 5 years only with 3 persons that you know, and sometimes you getting aggression, and this kind of things also happens. Osteopenia, visual impairment, as they mentioned, there's visual disc edema, that and they hepatitis, and maybe there's intracranial pressure increase that induce this kind of changes, and also radiation are exposed, because we know when you're going leave from Earth, radiation, you're totally open to space radiation. It's totally different, and risk of increasing cancer and reproductional problem is a lot they have to experience, and every astronaut have to train, have to take a medical training, because when you go into space, you contact them with Houston or Moscow, and they say we have a problem and doing some kind of precision, and they saying to you not stop doing that. But if you know, if you go into the Mars and other planets, this will take about 23 minutes to delay in communication, and this make huge problems. So this interestingly, and let's talk about Kazakhstani space. I will take about 1 minute, and this will be over. Kazakhstani aerospace industry also doing kind of research. And you can see from these researches, they're doing research about radiation, influence of radiation, gamma radiation on the psychological issues of human in space. They made experiment on the 4th astronaut of Kazakhstan, and this professor, they research, they have a findings. This Professor Sharipov, I meet him, and Professor Galina Davidovna from Institute of Ionosphere. And the problem is they have experience. They find making some scientific findings, but they have a problem. What kind of problem? They don't have a language, English language. They don't have international network. And I think if there's some guys pursuing in the future doing this career, aerospace medicine, maybe NASA, maybe another European, maybe Russian system, you can ensure that you making some research with them, with these guys. And there's the mail. If you're interested, you can contact with them. They have a lot of findings, but not printed internationally. And let's talk about extreme environment. There's beside of space, we also have aviational medicine, altitude, diving medicine, temperature, and all of them extreme environment physiology. If you're interested in this field, I think in the future this field will be very relevant. There's King College London offer a very interesting, very brilliant course of human applied physiology and aerospace medicine course. Nicola, can I talk for 2 minutes more? Yeah, thank you. Yeah, there's some textbooks you're interested. Very good textbook, aviation medicine, space medicine textbook of King's College London. If you're interested, you can look for this book. And there's some association. You can go and Google it. They offer some brilliant opportunities for younger generations that have passion on that. And also our neighbors. Russian Roscosmos, they have plenty experience in this field, but unfortunately, they don't have, a lot of people don't know English language, and they have a lot of findings that, for decades they have collect. And you can see the Institute medical biological problems. If you're interested, you can just go to the research gate and find some researcher and doing what they're doing and look for what they're doing actually. They have a lot of research projects on this field, like the space medicine. And if you have opportunity going to Moscow, you can visit this place. This is central training center of space for space astronauts. It's have a brilliant history. You can just visit this place, very enormous place. and you can go this place for training. And let's talk about latest news in space medicine. 2 weeks ago, SpaceX Dragon leaves the station, returned to Earth with valuable science. What kind of science they come to the Earth? First of all, they made some experiments of printing human organs in space, because there's a microgravity that totally you cannot experience on the land. And also they made some research on stem cells of heart tissues, and so some kind of biofilm festival, like microbiological researchers. It's very interesting. And let's talk about next month. We will experience new launch of SpaceX Crew Dragon with a human on board. This will be very interesting. And after that, maybe NASA will not use shuttles, and will use SpaceX companies. This launch is very modern, and it can seat about 3 astronauts. As we know, Soyuz can take only 3 astronauts, but there is 7, and this totally can encapsulated with International Space Station. You see that, and if you're interested, you can Google it. And what's interesting, we will experience in the future 5-10 years another space race, but among a lot of players. There's not only like 70s only America and Soviets. There's a lot of countries that will pursue this commercialization of space industry. And you can see a lot of rockets of SpaceX, Blue Origin, and also Boeing also doing this kind of research. And they will, after some years, there's a lot of bang in this area. And if you're interested, if you will prepare it for this, if you're passionate for this, you can study this and be prepared for working in this field. And let's talk about some problems that we have in Kazakhstan, and maybe in other countries also have this kind of problem: ecology and space exploration. We know that, first of all, to make some space exploration, you have to ensure that your bioecology and your peoples are safe. But you can see this was conference last year in Astana, and this was about modernization of Baikonur Large Complex, that to attract more customers in a future commercial space exploration. And there's the foreign brochure. You can look, if you're interested, you can download it and look for who is working. And there's some people in ecology and space exploration. And problem is, what I want to say, in Kazakhstan we have Kizlorda region, Jeskazgan region, and unfortunately we have experienced some parts of space shuttles that going on in villages, and maybe some people experience it like the kind of chemical toxic things that going from shuttle and just making huge impact on people that living there. And my point is, if there is some passionate persons that want to solve this problem, help to solve this problem, you can contact with these people and doing research on this field. Yeah, and lastly, I want to ask, what is the astronaut most want after arriving back on the Earth? You can consider about 10 seconds. How do you think what he actually wants to do? Everyone. Yeah, please.
Shower and a Coke. Coca-Cola, I mean.
Yeah. No. Just take a shower, because they experienced it in the space station about 1 year. They don't have a shower. They just using towels for taking a shower. And,
sorry, I was cheating. I saw the next slide, so I'm sorry. But it came to my mind in the same time as I noticed that, so I'm sorry.
So I talked with another astronaut. I watched the video of the astronaut, and he said, I want to take hours and hours of showering in the bath after come back. And if you have any questions, you can later ask about this field, because we humans, we have genes that have to explore more and more. And there's a good quote that says, our greatest accomplishment cannot be behind us because our destiny lies about us. And we have to be prepared in the near there's some resolutional development. So this is my Twitter, this is my email. If you have a question, please contact with me. Thank you. Thank you.
Thank you very much, Kalamjan.
Yeah. Yeah. This one.
Okay, that's great.
Thank
you.
Okay.
So at this point, we will have some time now for discussion, and we'll try to get through as many of your questions as we can. Unfortunately, Yerlan has been unable to rejoin us on Zoom, but he is over on Facebook, and I'm sure he'll be happy to try and answer your questions there. So perhaps I can address the first questions to Mukhit. One moment. So there is a request to please show us again how the nanotech vaccine could be injected into a cell.
All right, so which means that I have to share the screen again, if you don't mind.
Yes, please.
Yeah, so I took only like 5 or 7 minutes, so I think I have this luxury to use more time, right?
Yeah, that should be okay.
Okay, sorry for repeating this again. So it's always nice to see this slide because it took a while to do this. I'm not very good in animation, but I try to make it more visible because we are visuals and we understand better if we see. So again, the idea is to use the membrane or the envelope of this virus in the same time removing the inner part of the COVID coronavirus. So or else we can just we can use the translation or that's just synthesis of these viral components, as we already know what genome parts of this RNA is responsible. So any of these approaches can be used. One of them is to remove the genome, or the second is just to produce the membrane without genetic material inside, as you can see here from the previous slide. So after the entering the virus into the cell in cytoplasm, replication and the transcription can happen. So replication, we can eliminate this part only using this part. So translation of viral proteins and generating only the outer part of this virus. So any of these approaches can be used, and the idea is to use any agent. As I say, agent, it can be therapeutic or preventive agent. It can enter to the inner part of this membrane. So we call it membrane agent. This is the structure that can then enter the cell. So the question was, how can these drugs can be entered? So as I say, this membrane as a transporter for these drugs. So it's simply transport to the, we have lots of different types of cells with different types of antigens. So these transporters can find and recognize only those cells which are infected because of this membrane specifically created from coronavirus, and only can recognize these specific receptors, not going, for example, it doesn't go to muscle cells or the neuron cells which may not have these receptors. It will go to, for example, B lymphocytes or the lung cells which are vulnerable in this COVID-19 infection, and only can find these cells. And then after the entering, we call it endocytosis. After the entering to this cell, surprise, there is no RNA to be replicated or to be translated. There is only agent, only the drug that can remove and also destroy these viral structures. As I said before, it can be enzyme, the small molecules that can be implemented into the structure. These enzymes can be protease, for example. Protease is the enzyme to destroy the protein, or the nuclease, the enzyme to destroy the nucleotides, or the other, for example. It can be also the genetic material, for example, which can stop the replicating viral RNA, or which can stop the translation of viral proteins. So any of these things can be used to stop this life cycle of virus, as well as to destroy already existing components and viral particles inside of this. I hope I answered this question.
Yes, thank you. As a follow-up, perhaps you could address how you would remove the RNA from the virus.
Yeah, I think I partially answered this question just because I've read this question already. And again, it can be removed. There is already existing methodologies that has been used in adenovirus or other types of viruses, retrovirus, of removing genetic material from this envelope. Or else the second approach is to synthesize, or is to produce the membrane without the genetic material. We use the only transcription and translation part using ribosome and other peptides in the cytoplasm that can generate only the membrane. So I'm sorry, I'm not a pharmacologist or virologist, but still have some basic knowledge from medical school. So possibly it can be, sometimes some ideas are very simple, but some simple ideas can make some big changes. I hope this will help some people who are working in this field.
It's true, sometimes the simple approach is the way to go. Do you know if there have been any attempts to make this kind of vaccine in the past?
Yeah, again, I'm answering in the following up questions. Yeah, adenovirus is one of the biggest viruses already existing. It has been broadly used. Their particles, especially their envelopes, has been used for some adenoviral treatment as a transporter again, or the vaccination. So the main idea, for example, let's say someone has invented the drug or something, and these agents will follow and find the infected cells only, not, for example, affecting our neural cell or the blood cell, for example, red blood cell or white blood cell or any other cells. So we have to ensure that this agent, this drug, is very, very toxic for virus, but in the same time, it shouldn't be toxic for human cells. So we can put this into this envelope, and this envelope will go only to the cells which were already affected. Maybe we will lose these cells, but we will ensure that this virus will not be proliferate, and there will not the number of them viruses won't be increasing. So this is the idea, that we can use the transporters for already existing drugs or already making them. So yeah, this is the answer, I guess.
Thank you.
Could I add here a little bit?
Yes, Daulet.
As I understand it, Mukhit, you're suggesting the idea of a transport that, but not what will be inside. So it will be some targeted transporter, and to prevent the replication, propagation of this virus, and make it localized. Right?
Exactly. So as I started my presentation, that I'm not going to suggest any idea what we can use to treat or vaccinate people. I'm going to suggest what we can use as a transporter. Maybe it's the second question. I mean, first of all, we have to invent. I mean, anyway, we will find some sort of things that can possibly stop coronavirus. But the second question can be, how can we make sure that these things can approach only the infected cells? And I know the nanotransporter or microtransporter industry is highly studied. So previously we were using, for example, some liposomes or some other molecules, but for example, some hydrophilic or lipophilic sort of things that can ensure enters through some barriers. So in this case, these transporters are not just molecules that they're covering these drugs, but also they are covering, first of all, but secondly, they have these antigens, as I mentioned before, for example, MS proteins that can tropically find the TMPRSS and the 2 and the H2 receptors in human cells.
Thank you very much, Mukhit. We've got a question in for Daulet. What do you think about early diagnosis of the kallikrein, sorry if I mispronounce this, and other biomarkers in the blood, as well as neuroinflammation cytokines?
Okay, fantastic question. Be honest, it's what we are also doing. It's not kallikrein, it's the cariotherin, the appropriate name, the protein which we are investigating. So not a blood. Now we stopped on a cerebrospinal fluid because the anatomic region which is affected in a nucleopathy is the brain, and obviously the first liquid where it will be exposed, it will be cerebrospinal fluid. So we are approaching that we could take cerebrospinal fluid and detect the level of cariotherin and make a comparison between healthy one and a patient who is suffering. And it's a hypothesis, but we can say that it could be good biomarker to show the not just the existence of disease, but also the stage of the disease and how it's developing even. So it's a fantastic question. Yes, we are trying to apply cariotherin also as a biomarker of disease, also to identify the stages. Maybe in the future we'll use the blood, but first we stop on a cerebrospinal fluid. Thank you. Fantastic question, be honest.
May I also have question to other speakers?
Of course.
Yeah, so as we are speaking with Daulet, I have 2 questions. So the first question, are you planning to do some experimental research, for example, as you mentioned, for example, tau protein or beta amyloid? I've been using 3TG mice model for the Alzheimer's diseases. Possibly it can also help to your research. The second question is as I understood, because of this interruption in my internet, so you're looking for treatment target. So let's say you found the treatment target for neurodegeneration. So what the next stage? So what drugs or what things are you going to use? Maybe it can be the growth factor or any other things. Thank you.
Okay, thank you. First question is what I did now, because I'm on the beginning. Now I use my transgenic flies, Drosophila melanogaster. We use already existing model, but it's a fantastic model. We are very grateful to the scientists who generated them. But now I'm chasing to generate new novel transgenic flies, which could be comparable. So before starting this pandemic, I already finished half of my experiment. I generated new plasmids. It's like one step before generating your new transgenic flies. And hopefully after the post-pandemic period, I can continue my experiment, finish my plasmid. It's called subcloning, actual technique. Finish my subcloning, generate new flies, and now I'm stopped on the experimental animals will be only flies. It's more convenient to my experiment because they're living shorter and at the same time give enough amount of them. Mice is quite expensive, you should know it, and it requires a lot of energy and time. The same with flies, but it will be faster, relatively. About other proteinopathies, you mentioned tau, you mentioned CDP43. Yes, in future we have an idea, but it's not like now and all everything. Step by step, first nucleopathies. If we prove it, we move to the next group of diseases. Step by step, we can expand our borders and prove the importance of cariotherin, not only one group of diseases. Second question about targeting. Yes, it's a very optimistic. There should be lots of scientists all over the world chasing this, trying to find some protein which answers all questions. And maybe it could be potential target, and only way to manipulate with them is only pharmacological way. So it's generating some molecule which can interact choreofurin or interact the part of genome which is expressing the choreofurin, overexpressing it. By this, it's preventing the aggregation and the whole pathological process, or even can slow down it. And just adding a little bit to this question, as I mentioned earlier, it's not just treating, which is more realistic, it's also finding some diagnostic tool, elaborating diagnostic tool of all the early harbingers of this disease. Once we find it, it will be much more easier, because all neurodegenerative diseases, the motor symptoms or any clinical features start, cognitive deterioration. There comes after the pathological process. So it means that the process started, and it could be 40 years healthy man or woman who didn't even, just she or he is living a perfect life, healthy life, and not even suggested that he or she has these diseases. It's already started. And this disease is very, very smart in some way, because it's waiting when it's reached this clinical threshold, when it affected lots of neurons. And once it's affected, when it manifests in a patient, there's no way back. We can't treat it. As I mentioned, nowadays, we are not treating them. We are just supporting them. So there's like two approaches, diagnosing and treatment. But first, we must find the underlying mechanism. Sorry for the long answer. Try to be short.
That's very interesting. Thank you very much, Daulet. Okay. I'm just checking the other questions that we have here.
May I ask the question while you're checking? So, Mike, I have some question. Well, one question to Daulet and a lot of questions to Kaiyrzhanov, to Rauan. Rauan, so we all see that this incredible amount of funding that you are getting for your research, and you mentioned about the help or support of local governments. So, how do you think about Kazakhstan government? Are we ready to help in terms of financial and informational support to your project?
Thank you for the question. I think as I mentioned, we traveled almost to all of that countries in the region, and we met in some countries. We met the representatives of governments, including Kazakhstan, and I think in that region, the only country has been conducting some genetic studies and planning to do populational genome exome sequencing is Kazakhstan. And I think Kazakhstan would be our main partner and probably hub for these genetic studies in Central Asia. Thank you.
Thank you. We have a question submitted for you, Rauan. Please, could you clarify if some ethnicities have a stronger immune system to defend themselves from COVID-19?
There are certainly individual differences in immune response, but I'm not sure if there are inter-ethnic differences in immune response. But the current evidence suggests that some genetic factors can predispose certain populations to be vulnerable, susceptible to COVID-19. And the nice example of this is ACE2 receptors, which is angiotensin converting enzyme to receptors that we all have on the surface of our cells. And certain variants, I wouldn't say mutations, maybe certain variants have been shown to predispose to COVID infection and to death, to mortality. And there are 3-4 mutations that are more frequent in European populations and are very rare in Asians. So these factors can make a background for inter-ethnic differences between nationalities. Yeah.
Thank you. And you mentioned that there are some variants which may be considered a mutation for one ethnic group, but are actually a benign variation in another ethnic group. Are there any cases where these are pathogenic in one group but not the other?
Are you asking about ACAT or overall? I mean, in general.
In general.
In general. Yeah. For example, there could be mutations that are specific only for one population, and like a founder mutation, which is common in one area, but it could be absent, either it could be very common in other ethnicities and not associated with any disease. So there are certainly many mutations like that. So our aim is to find these founder mutations or emergent mutations in our region. For example, our research showed that in Kazakhstani population, we found emergent mutations that were already found in Pakistan, in India. Is it because we are kind of related? Well, so we want to explore this.
That's fascinating. Thank you very much.
Thank you.
May I have questions to Ghanemjan?
Of course.
Dr. Ghanemjan, I hope you should have taken notes, so because I have 4 questions, I'm sorry. Okay, I can ask just one by one so you won't get confused. So you mentioned about, well, not mentioned, maybe in your slides, I've noticed about epilepsy. So is there any differences in pathogenesis of epilepsy in the space and in the Earth? Maybe the question is huge, but maybe some big differences you can emphasize in these terms. So.
Thank you. Can you hear me?
Yes, we can hear you.
First of all, we do not allow, not we, but in this field, they do not allow
to fly.
Humans that have epilepsy disease to go to this field in space exploration. First of all, they just take off this kind of pilots that have this kind of diseases. But actually, I didn't think much more about these things, that epilepsy pathogenesis and correlations with space, microgravity, and maybe different influential factors. Actually, how I come to this field? I come to this field because our hospital in China, in Beijing, is one of the biggest neurological hospital, and at the time we have and a lot of people from whole China and difficult cases coming to this hospital and to finding some kind of hope, last hope. And at that time we handled another interesting case. That was a person that totally amputated from the level of the navel, and he survived. It was interesting car accident in Shenzhen about 15 years ago, and this guy survived. And I was really interested. And the issue that he came to our hospital, our clinic, neurosurgery clinic, there was phantomic pains that he experienced. That kind of he don't have a toes and lower part of his body, but he has this kind of experience. And at that time we determined to use spinal cord stimulation, and the pain was managed. And after that, I was really interested, because this kind of procedure, when I was looking for literature, this person was one of the third person in the world that experienced this kind of situation. Hemicarparectomy. Yeah, this surgery called hemicarparectomy is often used in a cancer patient in America about in last century about 70 years. And what interestingly, when you just cutting a person on half, you have to experience a lot of physiological changes, like temperature regulation, because you kind of skin, you don't have a lower part of skin. Yeah, how you will evaporate this heat, and a lot of field restriction and this kind of things. And after that, I just go into the Moscow, attend this kind of conference. Maybe there's a little another explanation, this kind of things. And by this kind, I was meet with this field, aerospace medicine. And so to sum up, I don't know about epilepsy correlation with microgravity, but maybe in the future, this is also a good field. Yeah.
Yeah, the second question, as I am ophthalmologist, I'm highly interested in eye-related, vision-related disorders. Yeah. So, yeah, you've mentioned about the disc edema, so possibly due to the change in the procranial pressure. So, I'm also I'm doing glaucoma research, which is related to intraocular pressure. The vision-related changes are inevitable in this space. So, how do you think about the IUP changes, intraocular pressure changes? Maybe it can be reduced or it can be increased. Maybe people who has glaucoma can fly there, and then I'm not going to use the flights to treat the glaucoma, but yeah.
Yeah, Mukhit, you mentioned a really interesting question. As space industry, it takes about for development nowadays from 16 about 50 years, and a lot of pilots. American pilots mentioned that when they experience long duration in space, they have a kind of vision disturbance, and but unfortunately, Soviet astronauts don't have. And after that, American health professionals, after every mission they come, they have to do some research on what kind of differences they have, and they mentioned one of the cause of human, and they found that there is edema of
optic disc.
Of disc. Edema of disc, and they just hypothesized maybe correlation with intracranial pressure. But I see a few days ago, I see NASA's research that they made, and also one student of master's degree, they made research on this field. She said that not totally correlate. They have correlation between two. There's also a lot of another things that affect this kind of condition, pathological condition. But so nowadays they offer like let's do to prove this kind of hypothesis. Let's do like some kind of regulation and make like let's monitoring this intracranial pressure. Is it really intracranial pressure or not? So this is an interesting field. I think in the future, and also they have to do a lot of research in this field. And if you're interested, Mukhit, as ophthalmology, this I think is a very interesting field.
Okay, the last question. Do you plan to visit aerospace? Do you plan to fly to the cosmos?
Actually, I'm 30 years old, and I take my bachelor degree in neurosurgical residency, and I'm just looking for myself or what field I want to develop in the future. And I feel that I'm pretty interested in physiological changes in human body. And if my vision is really problematic, and I think that at that time, if they develop some kind of condition, I really will try to experience this kind of thing. But 1 year not taking baths, this is a real big problem. This kind of you're living in a two rooms, the house that have 2 rooms, only 2 rooms. Yeah, and you experience 1 year.
Well, you can survive this COVID pandemic in aerospace and can be self-isolated in spacecraft.
Yeah, this idea, I'm interested on this kind of things and researching this kind of things. Yeah, I hope.
Kaiyrzhanov, perhaps I could add a further question. I was wondering to what extent we can model the effects of space travel on human physiology from the ground, because that seems to be a very rate limiting step in terms of the research.
Nicola, excuse me, can you repeat the question?
I'm wondering to what extent we can model the effects of time and space on human physiology from the ground, rather than needing to send people up to see the effects. I mean, obviously that's better, but that is slightly rate limiting at the moment.
Yeah. Really good question. A lot of things we can do in laboratories. There's, as I mentioned, the Russian Institute of Problems, biomedical problems. They bring this kind of research that we can do on the ground. And another NASA doing European Association European Space Agency, but kind of microgravity. But as I know, microgravity, you cannot create this kind of environment, this microgravity environment. And some research they made only in space, and want to look what kind of changes of these diseases can be found.
I guess the long-term effects of time and space as well is something that you would struggle to recapitulate.
Yeah, Nicola, you said about I remember that nowadays they're doing isolational experiments. It's named series, about 2 years of isolation. Maybe some crews of 3 or 4 persons are only in one room, and they're doing this kind of isolational experiments. But we have to handle this question because as Mask, if you go into the Mask website, Elon Musk website, they plan that in 2024 they have launch of human mission on the Mars. But if you're going to long way, the take for Mars will take 1 year, and take back 1 year totally, and you stay over there. And we have to handle these problems before they will go.
Yeah.
Yeah. Okay.
Yeah, that's a really big challenge.
Yep.
Go ahead,
Daulet. Galunjan, I think it's going out of our scope that space is an isolated environment, and it's also not challenging physically, but because it's also challenging mentally. Because I have, out of my experience, I was in one expedition, and it was a quite aggressive environment, and the isolation when you're not talking with no one. You are just it's a very difficult when there is no sounds, nothing. You and your thoughts, it's a very challenging, and I think it should be always in our scope. Everyone who want to investigate, in space, anything which is related to human, I think it's a crucial, essential.
Yeah.
Daulet is mentioning. Sorry. Yeah, Daulet is mentioning about his polar trip to Antarctic. So it's a huge achievement. So this is the dream. So one of dream to visit Antarctic, another dream is to fly to the space. So yeah, guys, cool.
Next time I will go to with
my plans. It's my next destination.
Yeah, and so the idea is like we have this kind of opportunities in Nazarbayev University. You have passionate guys, Kazakhstani guys, and maybe from other countries, and you can collaborate with Russian colleagues. And unfortunately, some of them don't have a language. You can use this tab that they collect for several years, and make this analysis, take the analysis this step, and find some findings. And we can do it. In my vision, just create some group of 5 or 6 persons, collaborate with that, and you can do our experiments on our astronauts. Maybe, as I know, our astronauts it will be launched from Baikonur after 2 years. Yeah, Kazakhstan, and we can do on them. Yeah.
Thank you. This is a very interesting discussion. I see we have a question posted for Daulet. I imagine you may have started answering this, but so the question is whether there is any research on the correlation between dietary preferences and neurological disease.
Yeah, it's look like I opened this function Zoom. I also seen this chat.
Yes.
Be honest, there's a lot of researches about correlation, not only neurodegenerative diseases, but nutrition and lots of diseases. But especially about neurodegenerative diseases, they're very controversial because, for example, in France, there was big randomized research and they finalized with ideas that, for example, areas where the traditional meal is a fish or any seafoods, the people who is living in these areas, they are more keen to suffer with neurodegenerative diseases. Meanwhile, in Italy and in Japan, they conducted another research, and the conclusion is vice versa. They're saying that no, seafoods actually preventing it. And I think it's a very intuitive. It's obviously everything is healthy in a appropriate amounts. even the alcohol. I'm sorry, I will be populist in here, but everything, anything could be healthy. And even nutrition, if you take it in an appropriate amount, it will be helpful. So answering the question, yes, there's a lot of researchers, and this is a sea of researchers, and you can jump in and find lots of stuff. At this point, what I read, I say that they are very controversial, but one conclusion, yes, there is an impact, but is it good or bad? No answer still. I also see that she asked two questions, Azure.
Yes.
Genetic basis, yes, absolutely. The Parkinson disease, which we are now investigating, Parkinson disease, dementia with Lewy bodies, they're all familial cases. It means that they have genetic basis. It means that someone in family suffered. Actually, it's also answering the next question, the third one. Person has history of disease in the family. Yes, if person is someone, any relatives who suffered with neurodegenerative diseases, yes, you are, I can say, in a group of risk, because these diseases has genetic basis. They are hereditary. They are transfer from one generation to another one. But the percentage is not so big. For example, in PD, it will be only 5% of all PD cases. So, and lots of cases, the genes, they are autosomal recessive. It means that it should be very rare case where the mother and father will be older, but not suffering with this disease. And when it's crossed, only one of the progeny will suffer with this NDD, while others not. So this is why it's a very rare cases, but we always should remember that we have this risk. If someone have in relatives, yes, they are definitely in risk. This is why, again, diagnostic tools will be quite helpful. These people can go through the screenings, like nowadays governments introduce oncological screening tests. We can do the same. And yes, I'm sorry, this will be answering too big, but that's it.
Yeah, and also, can I add just one comment to this question?
Please, go ahead.
Yeah, Daulet has nicely mentioned that there hasn't been any firm evidence in terms of correlation between food and neurodegenerative disorders. But in terms of Parkinson's disease, there have been some suggestions that dairy, so that we consume, like milk and other things, can predispose to Parkinson's disease. But this is just at the level of research. And also diabetes, type 2 diabetes, which is more common in Asians in comparison to Western Hemisphere, it can also predispose to Parkinson's disease. But these correlations, so in order to prove these, big cohort studies have to be done to prove, well, at least there are some hints. Thank you.
Thank you.
Yeah, sorry, I quickly scanned the chat and the Q&A section. So I've found one unanswered question directed to me. So, well, realistically, I would avoid to answer this question, but the question is that how realistic is it to do this kind of research in Kazakhstan? Do we have an infrastructure for that? It is a big shame that I don't have answer for this question. Well, if Dr. Yerlan Ramankulov would be here, he can possibly answer this question due to his background and the opportunities that he has. So as I mentioned before, I don't have a big background in virology or pharmacology, including pharmacy. But yeah, well, this is not the only fight of Kazakhstan or one person or one team against one virus. It is a fight of all humanity against this virus. I'm sure if we have the idea, we can find the infrastructure or other things that we need. So and we can achieve this only collaborating with our other countries, with other research labs, and with other scientists. So yeah, this is my answer to this question. Only interdisciplinary and collaborative approach can help us to fight against this virus. Thank you.
Thank you very much. I think that's important for all of our research, really, that collaboration and international collaboration is very important for progressing all of our research. I noticed there is also another question about using older techniques like breathing methods to help regulate the alterations caused by COVID-19 in an infected person. I don't know whether anyone could comment on that. Perhaps we don't have enough research to be able to answer this question yet. Mukhit?
Well, I'm not sure about the briefing method of Potato or other methods can help in that point. So again, I'm not a fan of the symptomatic or pathogenetic treatment. I'm more focused on the etiologic treatment. So we can win the disease only if we can win the virus, and possibly focusing on the things that we can maybe kill the virus or stop spreading it can help us, and can be the 100% and the radical treatment of this disease. But maybe if other colleagues have their own opinion, I'm more than happy to hear that.
Okay, so I'm not sure there are further comments on that, but we're just approaching the end of the session now. So I'd like to thank all of our participants for your questions. We've had some very insightful comments, questions, and also discussion from our panel of speakers today on our topic of healthcare and sustainable development. I will now invite the speakers briefly to share their final words on this topic before we end the session. So perhaps we'll start with Galamjan, if you have any final words.
Yeah, first of all, Nicola, thank you for sharing this kind of experience and being with us. I hope you're doing well in the UK, despite of this situation that's nowadays. And my last word, there's a lot of specialists that's giving speech about COVID-19. But I want to give some idea for younger generation of maybe Kazakhstan, Central Asia, that you have to think further in 5-10 years at what will be happen and prepare for this thing. And this field, as I explain, a kind of idea. You can choose if you're interested and make some useful things to help this to help insurance of humankind. Yeah. Thank you.
Thank you very much, Galamjan. Mukhit, your final words, please.
Again, thank you, Nicola, for moderating today's session. Thank you, Kazakhstan PhD Association in the UK. Very nice work during these 3 years, and for gathering us for the organizing these kind of events, for this network. I'm more than happy to be part of it. And thanks for the speakers for their nice talks and insights, for their answers and everything. And of course, thank you for the listeners and attendees. We are happy to share our knowledge in future. Please feel free to email us. And please stay safe, stay at home, and see you all soon after these shambles will be over. Thank you.
Thank you very much, Mukhit. Daulet, your final thoughts, please.
Thank you, Nicola, for fantastic moderating our today's session. It was quite interesting, and it was a good opportunity to see my colleagues and to exchange with ideas, to answer the questions. What I can say that we are living in an insane time. Everything is changing very fast. You know, stay safe, stay healthy, stay motivated, and keep doing what you're doing. I think it's the main ingredients. That's what my words. So thank you for everyone.
Thank you very much, Daulet. And Rauan, your final words, please.
Yeah, many thanks to everyone. I'm pleased to meet my peers from Kazakhstan, from other Central Asian countries. I think my final word would be, so let's collaborate. So we have to collaborate in research to be open. Thank you.
Thank you very much. I'd like to thank all of you once again for joining us today, to the speakers for such interesting presentations and discussion. I'd also like to thank the organisers for giving us this opportunity to share our ideas, because I think we've had such a wide range of talks today and different interesting ideas which are coming from our participants as well as the speakers. So that is all from us here today. I invite you to attend our next session, which is tomorrow, on energy and resource efficiency. Thank you, and please stay safe. Goodbye.